said that kind of give me a little bit of my background and already, and so we're
Which
gonna.
you would, yes, Craig. Yes, please. And tell us about Create.
Yeah, I'll do that. Yeah, just do. So we're gonna talk about some soil health things. I was I was worked for NRCS for 14 years, and I see Andrea Mann's here. Andrea Mann's here, and I used to work with her a number of years ago. But yeah, I used to work for Natural Resource Conservation Service, and then I got involved in the switch out of that and started doing started a business healing hooves for doing targeted grazing using goats. We're doing that for the last since about 2002, and been doing that for last I say that's a little over 20 years. And then I about a little over a year ago, I took the Create program, which I can't think of that exactly what that acronym but it's a ecology deep dive into soil health and coaching, and it goes into plant soil biology interactions and how we can work within the system to essentially work with nature to enhance uh, economic function, ecological function, maybe economic benefit as well. And so kind of balance those aspects of kind of work with nature to in order for us nature provide the inputs versus us providing the inputs. And how can we do that? And how can we work on creating that, like I said, the soil sponge, where the soil is working in a way that provides nutrients and absorbs the water and all those aspects of a well-functioning system. So with that background, I kind of go into what is the soil sponge, and kind of go into what is important and some ideas on monitoring that, and then some cover some ideas on enhancing your soil sponge and some what you can do to do that. And if there's any questions as we go through that, just like I say, it's a small group, so I don't interrupt or say something, raise a hand. I'm not Tom can probably see the hands better than me, but if somebody has a question, just holler it out, and we can answer as we go along. And we already talked about that. And so one of the things when I remember taking soil class, and that's in the 80s when I went to the University of Idaho, there wasn't a whole lot of talking about how the plants feed the soil biology. It was more of the chemistry side of things, and it's just a fascinating aspect when learning to create. I do kind of the plants and the soils are important, but I didn't have a really good background in how well the interaction between the plant and the soil biology is, and how the plant excludes root exudates and actually feeds them microbes in a way that they communicate essentially in a way through the chemical compounds they excrete, and then there's an interrelationship between that, so that the plants provide them with food, and whether it be bacteria or fungi or different aspects, they provide food back to the plant, and also some certain getting be more integrating, so how well they also provide some protection for the plant as well. So that is an interesting aspect of that, and the soil sponge is really that community of organisms within the soil that create that interrelationship between the plants and the soil biology. As you think about our community as a human community, there's highways, and there's hospitals, there's grocery stores. So there's all these different aspects of community. In that same way, within the soil, there's all that aspects of the soil as well. There's building blocks there. The bacteria provide kind of the building blocks. The fungi kind of glue those smaller building blocks together and form that structure. And then the hyphae, also the fungal hyphae, also bind that soil together. And so you got and then there's there's bacteria, there's like say fungi, there's earthworms, there's nematodes, and viruses. There's all kinds of organisms, and then insects and arachnids, and so there's all kinds of this diverse life and a well-functioning, healthy soil. There's all kinds of biology in there, and all these interact together to create a community. And that soil aggregation of the soil is important, as we can see when you're out after a rainstorm or something like that. If you get a rainstorm on a soil that you crust over, for example, crusts over real easy after a rainstorm, it's indicating that that's probably a fairly bacterially dominated soil. You don't have that structure, that is not held. Essentially, that structure dissolves when the rain hits that soil. So if it does that, then it closes off those pores, so the water can't be absorbed in, and it just sits on the surface of the ground. Whereas if you have that well aggregated soil, it acts more as a sponge. The water, it holds its structure because it's got the glues and the hyphae that hold that structure together. And then there may be also bigger pores from the earthworms and those aspects that are also provide spaces for the water as well as the air to move into the system. And so that when the water does come, it doesn't dissolve that, break down those, like say the small bricks. It has the bigger building blocks in there, so that it's like building, the analogy of building a building on sand versus stone, where you have a sand, it just melts away. Whereas in this situation, you have the glues and everything, the mortar that holds all this structure together, so it holds up against the rain and so that it doesn't create it doesn't seal over and the water goes into the soil. So it will increase holding capacity. It also increases enhances nutrient availability as well. And healthy soil, as we've just said, it's really, the soil is alive. Typical soil is made up of, this is just on averages. There's different percentages of organic. Main thing that, well, there's a couple of those things can change, but minerals make up 46 or roughly almost 50% of the soil matrix. There's air within that soil, and water, the pore space. That's 50% of that soil in a well healthy soil is pore space, and then your organic matters 3 to 5, it varies between, and if you get 3 to 5% or sometimes lower if you're in a cropping system. But that airspace, and one of the things that you think, well, what is the most limiting factor related to plant production in soils? And you think, somebody thought a thought. What is the most limiting factor for soil production in a soil system, in a soil? Somebody throw out something. A thought.
Well, you think it's water, but sometimes it's nitrogen.
Yeah, you get waters can be very limited, and
I would guess air.
What's that?
I would guess air.
Yeah, and that's air is critical. You think about it, how long we can go out without water for 3 or 4 days. Air, we can only go for a few minutes. So air is critical. It's really one of the most, we don't think about it as being critical, but it is very critical in the soil situation, in the soils, because that's, the biology is just like us. The biology in the soil breathes, the soil breathes. So air is really critical in that situation. And so if you got a situation where that if you get a soil that seals over, it's not going to bring in water, it's also not going to bring in air. So that impact has a huge impact on the availability of soil biology and the ability of that soil to function. Just a second here, let me. So anyway, those are some of so yeah, air is really a very important aspect. I mean, nitrogen, all these other nutrients are also important, but if you start limiting some of the things as you question, looking at some of these limiting factors, which ones are the most critical to start address as you work through the system? And then we just talked about that. What is less limiting factor? And some of those, think about sunlight. Sometimes, you don't think about sunlight being limiting, but if you're in a grazing situation and you're not managing the the grass, and you're not having and you're there's not much leaf surface area, then sunlight can be one of your most limiting factors. And then, like I said, mentioned air, water, decomposition. How is the nutrient cycling within that system? And then, as it was mentioned also, that macronutrients could be limiting factors. So all these factors can be limiting, but based on these particular situations and how things are being, how the land is being managed. And one of the primary aspects of the soil is the bacteria. I mean, it's interesting if you take a teaspoon of soil, you got 100 million to a billion bacteria in that particular soil, which you don't think about. It's just the soil has the amount of life within the soil is tremendous. There's been studies done that said that when the bison were on the plains, there was as much the biology in the soil was just as much as the bison on the plains. You don't think that would be possible, but there's a huge amount of biology in the soil. And so bacteria, one of the simple ones, they provide the building blocks, and that they start building some of them. They essentially are the bricks, part of the building blocks of the system. They are typically break down, they're decomposers, more simple sugars. When you're in when you're doing a composting or something like that, if you're the higher the carbon nitrogen ratio, and the more nitrogen in a system, that's when you get heat in there, because that's usually the bacteria that's going. If it's less carbon, that's when the fungal got breaking that down, and it's a lot cooler system because they don't generate as much heat. But they are the breakdowning simple sugars, and they are critical in the nitrogen cycles. There's several bacteria that are nitrobacter, important for converting different nitrogen to nitrates, to nitrate and nitrite. So those bacteria play a really critical role in the system. And there are bacteria that actually are, they'll solubilize phosphorus and zinc and some of these other minerals in the soil that are specifically making certain other minerals available. And then actually bacteria are very high carbon to nitrogen ratio. They're usually we'll talk about that a little bit more, but they're a key part of the soil food web, and they consider their bacteria, their carbon to nitrogen ratio is like a 5 to 1. So it's a pretty high, it says 70% nitrogen. They're very high levels of nitrogen. So they're like little packets of nitrogen running around in the soil. And talk about the nitrogen cycle and those various aspects. You get into the nitrogen fixing. There's actually I know most people know about the rhizobia, which is the bacteria that are in the form nodules in legumes. Those are actually held within the nodules of legumes. And there also are free living nitrogen fixing bacteria that just live within the soil, and also those are in areas that they have to have conditions within the soil that can limit some certain level limit of oxygen. So that's where if you have a well irrigated soils, there are pockets within that soil that has less oxygen than some other areas. So those pockets can be areas where some of those nitrogen fixing bacteria can live. So those nitrifying bacteria that convert ammonia to nitrates, and then there's other nitrifying bacteria that treat nitrates to nitrate. So then that plant makes a plant available. So there's a lot of roles that the bacteria play in the nitrogen cycle. And then we get into fungi, various aspects of fungi. One of the key ones you hear about a lot is a mycorrhizal fungi, which actually forms symbiosis relationship with the root system. They actually, some of them are on the exterior of the root, some actually go into the interior of the root system. And these are, they have hyphae are really, I mean, that picture showing there is the bigger part is actually the root hair, and the little fibers are the hyphae on the mycorrhizal fungi. So they're very small, and they can really, that's one of the huge benefits, as it says here, they can increase the surface area of the root hairs by up to 40 times. So they can really significantly increase the ability of that plant to access nutrients, because they can, and as also water, because they can go into areas where actually the root hairs can't go into, and they also, they transport water and nutrients. And I've seen, I didn't show it here, but I've seen videos where actually the bacteria will, the water, there may be not a lot of water in the soil, but there's water really adjacent to those hyphae, and so the active bacteria will move in that. There's a kind of a water column with it next adjacent to those hyphae, so the bacteria can actually move in the soil along those hyphae and they're providing nutrients via that pathway. And they do, I mean, like I said, that one of the things, mycorrhizal fungi, some of the different mycorrhizal fungi, I think they exclude glomalin, which is kind of a glue, which is what I mentioned earlier about gluing the bacteria, the bricks that the bacteria kind of put together. They glue those into buildings, and they build the structure. They hold that system together, and it doesn't, it's resistant to breakdown from water and other aspects, and it acts like a sponge. If you compress the sponge with your hands, and it'll come back. And that's same principle applies to the soil. If you have that soil that has a lot of biology in it, and you run over it with a piece of equipment or something like that, you may compress it, but it bounces back because it has that ability from the organic matter and the biology to bounce back from that, because of the structure that's there that has resilience that will bounce back from that force that's put on it. Next slide. And this is a picture of showing that rhizosheath that goes around the root. It forms around and within that root. If you have the one that second picture, which shows A and B, one A has very little rhizosheath around it, mycorrhizal fungi around it, and the other one B does. And that it's providing nutrients, but it also provides protection from that plant, again, from either because it's colonized that root system. So some other harmful bacteria cannot get into that, cannot access that root. So it's providing a barrier there. And there are different fungal and different bacteria. Some will actually produce, like penicillin is a bacteria. So they can actually produce antibodies, and so they have ability to provide immune system for that plant through the root system. So there's a lot of interactions going on there within that plant system. And this is kind of a funky picture. This is actually it's an electron microscope picture. It is that is a nematode, and that's a nematode is being attacked by a fungus. That is a fungus that has trapped that nematode and is actually going to will dissolve that nematode. I know there was, I don't remember don't have the particular example, but they were doing a study of where they were trying. The plant was they were using an insect to evaluating whether or not it was going to eat a certain fungus, but it turned out the soil was biologically active enough that the fungus ate the insect. Because there are certain funguses that will can break down the structure, the chitin, which is the makeup of some insects, so they can dissolve chitin. So it will actually dissolve, larvae and different things within the soil. It has the ability to control insects with, in that are living in or eggs or other insects that are potentially be harmful, that they can actually dissolve that chitin. And then if you're looking at, I don't know if you heard much about the bacterial fungal ratios, but you got the more simple, I guess, lower successional bare soil or situations usually are more bacterial dominated soils, and some croplands are that way too, because with the disturbances that have been applied to those systems, you have typically the fungal component usually goes out first. It's more susceptible to, well, when you were using some situation, we're using fungicides, and then of course tillage has a big impact on the fungal component. So how much those are used has a big impact on the ability of the fungus to survive. And if you, in most situations, in a pasture, probably a rangeland situation, you're probably like a 1-to-1 or 2-to-1 bacteria to fungal ratio. And then in some of our cropland situations, we can be, it can be like 100-to-1 to bacterial dominated soil. So, and then when you get into the forested situation, that's where, of course, people go hunt mushrooms. You're going to go look in the mushroom. You're going to look in the forested situation. Not usually, you'll sometimes find mushrooms in a range situation, because you're getting more fungal dominated than it would be a typical cropland. But your forested situation is highly fungal dominated. It'll be, again, if you look at those ratios, it's more the ratio goes the other way. It's probably would be 10-to-1 fungal to 10-to-1 fungal way. So it is a highly bacterial dominated, fungal dominated forested soils are highly fungal dominated soils, and that you're usually breaking down. Fungus is usually ones that break down more complex carbons. So they're breaking down the wood and the leaves and those aspects that are high of a higher carbon to nitrogen ratio than whereas in your other situation, bacteria are their ability to break down is more on the simple, like I said earlier, simple sugars aspect of things. And then, as we mentioned, people have heard of the soil food web. This is, when you're, as I mentioned earlier about the, like carbon nitrogen ratio of bacteria, it's like 5-to-1. And then like a protist or that eat bacteria are more looking at more of a 30-to-1 carbon nitrogen ratio. So if a protist has a 30-to-1, it needs 30 carbons, but it only needs 1 nitrogen. So it has to eat 6 bacteria to get to 30 carbons. So in that situation, it has 6 nitrogen, but it doesn't need 6 nitrogen. It only needs 1. So it releases those nitrogens into the soil system. And that's kind of the soil food web situation, where you're looking at those, like I said earlier, the bacteria are like little packets of nitrogen that are floating around the system, but there's got to be something there to harvest them. And that's where, if you have you got the food web, or you got protists, you got nematodes, and there are bad nematodes. There's bacteria and fungus that impact cropping system, that natively impact cropping system, but there are also beneficial ones that eat bacteria, eat fungus, bacteria. Nematodes eat bacteria. They also eat fungus, and they are eating some of the bad fungus and the good fungus. But it's kind of maintaining that balance where it's able to in a natural system, it's able to maintain a balance in most situations. There are situations where that doesn't happen. You get a disease that's happening in a forest. You get there's a stress conditions or something happens in the system that causes that to get out of balance. But that is, when they talk about the soil food web, is that how that system, that's why, the why there is the plant is it doesn't need outside inputs to provide the nutrients, is that you got the soil food web, and then there's bacteria that fix, free living bacteria that fix nitrogen. There's, fungus that can break down also like the phosphorus bonds that bind phosphorus and iron. Because one of the challenges with when you apply phosphorus to a soil, a lot of times that's bound up fairly quickly within the system. And if you don't, because of the chemical reaction, it bonds quickly to calcium and magnesium, and so it forms a compound that's not really accessible to the plant. So you need that biology to get access to that, the phosphorus and some of the iron. So a lot of times, if you did a total digestible and more of a mineral a mining assay of your soil, there's a good chance there's a lot in the soil, but how much is it available in the system? So this is just kind of talking about all about balance, and how do you, as we said earlier, you've got a left-hand side is more of a bacterial dominated soil, and you get the crusting going on because you don't have that structure that is holding that it holds together when it gets exposed to moisture. Whereas on the right-hand side, you got a more higher fungal component. You got that, so it's able to hold the building, soil aggregates, the building blocks are held together. And you got more pore space because you got worms, or they're creating pore space within the system, and they're cycling nutrients as well. And there's a couple of things about earthworms that's quite fascinating. I didn't realize that as well, but there are some aspects of earthworms that are pretty amazing. How the castings from the earthworms have a lot more available nutrients in them than just normal soil. And it's here, 5 times more plant available nitrogen. So they are able to break those, the decomposing plant material, and that makes available nitrogen. They're also doing that with some of the phosphorus as well, and some other nutrients. And one aspect of it, if you have a fair amount of earthworms in the soil, they're actually they're dead bodies, and that's the same way with other organisms within the soil that are called nectral mass. It provides a lot of nutrients, and earthworms are fairly high level, have a fairly high levels of nitrogen in their bodies. So they have 25 earthworms per cubic inch of soil. You're looking, roughly 25 pounds of nitrogen per acre due to their dead body, decay of their bodies as they die and in their system. So it is or having earthworms within a pasture situation. There was studies done that it can add up to 10 to 30% to the productivity of that particular pasture. So they are a key element in that in a healthy functioning soil. And as I mentioned earlier, we talked about there's our growth promoting bacteria, and the picture off to the right hand side is a alfalfa root system with nodules on it. So that's where the rhizobia live, and it's one of the interesting aspects of it that you can learn a lot about looking at and we look at a legume and during the growing season and just see how many nodules do you have? If you're looking at your system, how many nodules do you have? And then if you cut them open, are they, because they have, it's a globin, but it's not hemoglobin. It's a different globin. So it's kind of like our hemoglobin. It helps transport nutrients as well, but ours transports oxygen. But it helps. The color of the as you cut that nodule open, you can see if it's bright red or pinkish, that is actively producing nitrogen. If it's like if it's white or white, it's not currently fixing nitrogen. So you can tell, you can give you some indications of what is going on within your nodule if your legume is actually fixing, how well it's fixing nitrogen. And then the other picture is really just talking, looking at, it'd be all those microbial bacteria and fungal components, the root exudates are feeding those that soil biology, and the soil biology is in turn making nutrients and available to the plant. And there are also bacteria on the leaf surface that actually can also fix nutrients and also provide protection from diseases and that are protect the roots. Protect the leaf system. So there's a lot of things going on in the system. What's kind of the impact of a healthy soil? Looking at, as we've talked through these, increase nutrient availability and impact plant disease issues, and there are compounds that between the plants and the bacteria and the fungus that actually the growth promoting compounds, hormones, and that are produced that actually reduce stress. Plant stress, and it can help, which can make a plant able to better withstand drought or salinity conditions. So there's a lot of factors that play into how these plants and soil biology work together. And then I could just go a little bit into some of the monitoring side of things, as far as a lot of times people do a chemical test, but there are some other aspects you can do to looking at this, 3 parts of the soil, which is the chemical, the biological, and the physical characteristics of the soil. And what can those things tell you about what's going on within the soil itself? A chemical, you're looking at some of the things that you may want to look at is like the cation exchange capacity, which essentially the potential of that soil. If you have a sandy soil, it's going to be have a low cation exchange capacity, and its ability to hold, it's calcium, magnesium, potassium, and sodium, and hydrogen. So those are some of how much of those, what percentage of those are on. If you do a base saturation, another part of it shows you what percentage of those are on your soil colloids. So that gives you an indication of how that breakdown is, because that calcium magnesium ratio is important aspect. So if you get calcium more, it will make the soil more porous, fluctuates the soil. Whereas if you have if you have a lot more magnesium and calcium, it'll actually make a tighter soil. So those kind of things are important to know what's going on within your soil. And then some of these micronutrients, sulfur, zinc, and boron. The boron is one that, if you look at your alfalfa or legume, and or you can even see it in broccoli too. If you go to the store and look at the stem of your broccoli and it's hollow, it probably had a boron deficiency. And that's saying it's one of the aspects that would give you indication of what's can give you some indication of what may be going on with your plants by the characteristics of your plants as well. Some of the biological, there's a lot of different biological tests depending on how much you want to pay, I guess, is that big one on that one, because there's you can spend a lot of money on biological tests. The microbiometer, which I have one, it's probably about $100, and you can do 15, 20 tests. It gives you a bacterial to fungal ratio. It's a pretty simple test that you can do yourself. There's a Haney test, which does a kind of a combination of chemical and biological. There's a phosphofatty lipid acid test that gives you some indication of biology. Earthfork does a test that will give you total fungal and bacteria ratios, and if you want protozoa and nematodes, it'll give you that detailed information. They're getting, the Earthfork is probably in the 150, depending on how much information you want, $150, $200. And then the biomakers, they go into DNA analysis, and that's interesting. That it's one of those things that they can give you a lot of information, but trying to figure out what it all means is probably the next challenge, because they'll break it down into the functioning. Like I mentioned earlier about some of these bacteria that they may be say nitrogen soluble, nitrogen fixing bacteria, and they might look at the phosphorus solubilizing bacteria. They could tell whether or not those are functioning within your soil based on their DNA analysis. So you know that, then how do you fix that is probably the bigger question. Sometimes that's the challenging aspect of understanding that part of it. The physical aspects of it, just looking at the physical characteristics of your soil, understanding one of the things, of course, understanding your soil texture is important. You can do a slake test, look at, infiltration rates. That's probably a good one to tell, if you can't, do an inch, roughly the second inch of water within 10-12 minutes, you've got an issue with, infiltration rates. So what's going on with that? What's happening there? And then, the visual, I've done couple different visual soil assessments, which is, essentially digging a hole. Looking at the soil irrigation, you're looking at the smell, you're looking at for compaction issues. Do you have compaction? Do you see, do you find earthworms? You have capping going on. Some of these kind of characteristics are going on, and then you can do your soil, your underwell test. You got a 100% cotton underwear. You go out and bury it in the soil and see how long and then come back. Usually, I can't remember exactly the time frame, but it's like 4 or 5 weeks later. You come back and check it. Is it breaking down, or is it just the same way? It just looks dirty. Versus if it's broken down, then you got a lot of biology work because it's breaking down that cotton. You can't do it with, of course, with polyester or something like that. It'd have to be cotton. It'd have to be a cotton system, cotton underwear. And then the slake test is another step one the person can do. This is one I did a while back. It's conventional tillage situation where and then a no-till annual spring cropping, and then the arranged land situation. So this is showing you how that soil holds up with water. You want to collect that sample and let it dry for like 24 hours before you do this, because it doesn't work. You got to have it. It's got to be semi-dry before you do this, and then put it in there and see how it holds up to the water. And then again, it's those glues that hold that, or how well is those glues and those biology holding that soil together. A conventional tillage situation where you've got tillage, you've got that's impacting the biology. You've got various either fungicides, potentially fungicides and seed. We could have potentially some of the synthetic fertilizers can have a negative impact on soil biology. So all those factors are have an impact on that ability of that soil to hold together. And then no-till system, less tillage. You probably still have some of the synthetic fertilizers, but it's still with the less tillage, it's holding together. It has more irrigation. Then on the rangeland situation, you don't have that situation. You have a stronger, a higher bacterial, fungal to bacteria ratio, so it's holding that system, that soil together. And then some things you can do to you can monitor some plant leaf tests. There's some things a person can do. There's tissue tests you can do now. Now they're doing SAP tests, which is kind of the tissue test is kind of like you're doing a hair test. It takes a tissue test is they take the tissue send in a tissue and they just it's a destructive way of evaluating what minerals are in that soil. The SAP test is really more like a blood test. They don't you send it, you have to it takes more care to take a sample in the SAP test, because they're comparing old leaves and new leaves as far as the nutrients in those old and new leaves, and then how mobile nutrients are within that. If they're mobile nutrients and you have deficiencies, you'll see the minerals will transfer from the old leaves to the new leaves, so you see deficiencies in the old leaves. So because mobile nutrients and in mobile nutrients, you'll tend to see the opposite, because they're not really mobile, so they're not moved from the old leaves to the new leaves. But that takes a little more. The sap test is like, say, they almost pull the sap out of the leaves. They don't destroy the leaves. The leaves still the same. They just pull the sap out of them. I'm not sure exactly how that process. I don't know how that process works, but it is you have to send in a sample in a sandwich bag, and you've got to keep it cool so that integrity of that sample is good when it gets to the lab. And another test that a person can do themselves, if you want to buy a refractometer, is a Brix test, and that's kind of it really measures dissolved solids, and it gives you there's some charts a person can look up as far as telling you what how that, if you have the higher the Brix, really generally the better is a better indication of nutrient quality and ability of that. There's been some studies done on how insects attack different plants relative to the Brix levels. I mean, the Brix level is going to change throughout the day. Early in the morning, when the sun's, if you do it before the sun's up, the Brix is going to be lower than if you do it in the middle of the day, because it's actively, the plant is in the middle of the day when it's photosynthesizing. It's producing more carbohydrates, so it's going to have a higher Brix level, which it should have. So ideally, you would do this Brix test at the same, in a sunny, early afternoon, sunny day. You would get and compare those. It's that same similar time frame as if you were trying to compare Brix tests, and it can give you, if you have if the Brix test is down in the 3 to 4 range, you have to really low Brix. If you're getting up in the 12 to 14, you're getting a higher Brix level. I think for alfalfa or something, you want it to be, ideally it'd be up in the 12 to 16 level. If you get down and Brix down to 3 or 4, you can actually, that's when if you get aphids attacking a plant, you have you're going to have your indication of low Brix, because they're going after those simple sugars. And that's what they're able to, it's like aphids and those sucking insects, they're going after very simple sugars, and that's what they can digest. For some reason, those simple sugars are not being converted into proteins. Maybe in some situations it can be higher levels of nitrate, or it could be some other, it could be a micronutrient that's dealing with not an enzyme that's not actively converting that, available to convert that nitrate to a protein. So that just can give you some indication that something's not quite right, and then you may indicate what you need to do some more tests to evaluate what's going on. Some of these things are enhancing the soils run short term. Some of the things, it's really some of this disturbance, some disturbances has a negative impact. Reducing tillage, of course, reduce the impact on. Doesn't this mean that tillage is bad? It's just that lots of tillage negatively impacts fungal component. And then determine your enabling factor. That was when we were talking about, what's your limiting factors? Is it sunlight? You're managing pasture. You're managing adequate recovery, those kind of things, or is it air? Is got dealing with compaction issues? Are you doing how well is your material? If you're in a cropland situation, how well is the residue decomposing? If you're in a pasture situation, look at your manure. Is it breaking down? So some of those things that are going on, and then the macro or micronutrients can be an issue. And then again, address chemical deficiencies. Sometimes they're deficiencies. Sometimes excesses can be worse of a problem than deficiencies. If we get too much of a product, too much nitrates in a soil. I mean, I know that's poisonous to animals. Can be too high levels of nitrates can be an issue, and that can also be a problem for the plant. If we get too high, then you're not getting those nitrates. It essentially dilutes the carbohydrate, dilutes the system, because it brings in more water, and then you have a lower Brix, and you're going to attract more insects in that sense. So that's another balance that can be a challenge. Some of those fungicides, of course, are going to impact fungus. There's some biological seed treatments out now that work well, and then one of the challenges with synthetic fertilizers that there's some of them are impact biology. If you have high levels in some situations, essentially, if the plant is getting all of its nutrients from the synthetic fertilizers, then it doesn't really need the biology to provide those nutrients. So it's there's a give and take there. So if it doesn't need nutrients from the soil biology, then it doesn't necessarily feed the soil biology. So then that impacts the soil biology in that sense. And then if you're in a pasture situation, eliminating overgrazing so that you're creating able to actively growing plant for a longer period of time. And then positive steps, again, in a pasture situation, you're looking at providing adequate recovery periods after grazing, managing the leaf area that's available so that you have a actively growing plant as long as possible. And then there's always a correlation between above ground growth and root depth. So you got a short plants above ground, they're growing short plant above ground, you're going to have a shallow rooting depth, which in turn will impact your plant's ability to access nutrients as well as water. So are you creating your own drought situation? And then you're looking at some of those infiltration issues. Is there a compaction issue that you need to address too? That could be an issue that either, and especially on a cropland situation, it could be due to tillage. There can be a tillage compaction layer. There could be, chemical. It could be the compaction layer. Roots can go through roots can go through amazing. I've seen roots go through pavement and concrete, but it's what the compaction layer does. It restricts oxygen in those layers, so you're getting alcohols and other aspects of formaldehyde and different things that are in there that are toxic to the roots, so they won't penetrate. They won't go through the essentially the poisons that are in that anaerobic zone. So you've got to figure out a way to to break up that. Some plants have a higher ability to do that than others. And then how long you can keep an actively growing root system. The longer you're growing root system, the better. More it's feeding biology, and there's been a lot of work to provide on cropping areas, putting down they're putting down sugar molasses, they're putting down different compounds, they're providing sugars to the biology. Well, the most efficient way to do that is to the plant itself, because they can do a lot better, more efficient system way of doing it than we can, a lot less costly way. So then having a covered soil, of course, so you have the food for biology, and one of the biggest thing I think is I was talking to somebody today is about providing shelter, because the biology needs shelter as well, because they have to have cooler, they may hold moisture in there, as well as moderate temperatures, because if you get better soil, you can have hot temperatures, and if you get once you get much above soil temperature of 100 degrees, you start impacting soil biology, and so that able to keep that soil cooler and moisture is a huge asset for soil. And then most people have learned the 6 principles of soil health. They've been around there's either 5, you see 5 or 6 principles. Now they've added context, which is essentially meaning how these principles apply to your specific situation, because they're each farm or ranch is different. How does it fit their economics, their environmental conditions, as well as the social condition, the family and interactions there are all important in how you apply these principles, and how can you work with disturbance, work with nature actually. And then sometimes it says do not disturb, and I think that's because one of the things is probably there's certain types of disturbance that are like lots of tillage is not good. But some people do a severe grazing, which is not necessarily a bad thing. Sometimes that is needed if you're trying to put residue onto the soil surface. You may want to have a high level of animal impact in that kind of situation, but then you got to have adequate recovery to make sure that the plants recover from that prior to grazing it again. So it's how the disturbance is managed, I think, is more of do not disturb. It's more of how the disturbance is applied and what type of disturbance it is. And then again, the soil cover, and as we said, that again, you're fighting surface armor, soil cover, and diversity is big, whether it be diversity of livestock, diversity of in a grazing situation, or diversity of plants when you're doing a cropping system. because that diversity feeds diversity of plants feeds diversity in the biology, and nutritional qualities of different plants have different nutritional quality, and that impacts livestock. So all those different things interact together to be a benefit to nature, and keeping the living root system. Again, it's feeding the soil biology, and then incorporate. Now it's looking at incorporating livestock onto cropping system, because it's adding that another layer of biology of cycling the nutrients back into the system. So it's an interesting aspect of how we work within how to manage in a complex system, because one of the challenges with this is there's no, it's not a simple thing to do. It has a lot of complexity, and managing complexity can be a challenge in itself. And the soil sponge, it's just kind of a summary, is it's looking at how we're managing those four ecosystem processes. How is that, looking at the water cycle, how well is it functioning? How well is the nutrient cycle functioning? Yeah, the energy flow, how well are we collecting soil? How well, if we're grazing the site, overgrazing the site, or animals are grazing the site for extended period of this time, that ability of those plants to harvest sunlight is limited. How long is in a cropping system? How long is there actually plants there that are growing, that are harvesting energy off that system? So if you get an annual system, you may have a growing crop for only about 4 months. So how do you increase that ability, that the system's ability to capture energy? Solar energy, because that's about the only thing that's free nowadays is the solar energy. So how can you capture as much of that as potentially possible? And the community dynamics is really that diversity. How do you add diversity to the system? Whether it be in a cropping system or a pasture situation, get more diversity. Again, different plants have different rooting depths, different nutritional profiles, so animals can get different levels of nutrition from different types of plants. So it's all, and then if we can work within the system, and then we have to provide less inputs, and that can potentially help with the profitability and resilience of the operation. And that's pretty well all I have as far as presenting in this about that. I don't know if anybody's got questions, or we kind of talked a lot, but
Craig, thank you very much for presenting that. It's a lot of new information, and it's very helpful. That's nice.
You're welcome. Yeah,
I was.
Oh, go
In
ahead.
the Q&A.
Oh, okay. I didn't see the Q&A, but let me go find let me see if I can.
Yeah, I'll read it. It's in a
Oh,
rangeland situation, how do you determine your enabling factor?
In a rangeland situation.
Yes.
Well, it's probably very similar. You could do soil tests. You could dig a soil. Do you have, I guess the question, do you have compaction issues? Do you have, you could dig a hole, go out and dig a hole in the rangeland situation. How is how is your manure breaking down? Do you have gray is the system, I guess sunlight, first thing I'd look at sunlight. Okay, sunlight is what's the vigor of your plants? How well the plants growing, and what's the vigor and the growth of plants? I'd look at, that'd be one thing to look at. Air, is there any type of compaction issue, in your soils? And you can have compaction on rangeland. It's depending on our pasture situation. So digging a hole and check and see if you got compaction layer. Then water, how well is the water infiltrating into? You can do an infiltration test and check water and how well the water is. You can do that on rangeland just as you can do it on cropland. And then biology, you can do a biology test. You can do a soil test on rangeland just as you can do on the other system. It's just what can you do to fix that is the other question. It's probably more of most of those areas that most people don't apply biology on. People do have applied biology on rangeland to address situations. I've read about Nicole Masters' book, you'll read that the person did modified a old army truck, and so they could they were putting on I think worm castings onto and biology onto the rangeland, having an impact, a positive impact on their range production. So there are ways that can be done. If that's determined to be needed. Again, I would look more at the management side of things. Start with, because those are probably some of the easier things that you can change about looking at. Is there overgrazing going on? Is there what's the recovery like? Those kind of factors, looking at those situations.
I think a follow-up question, Craig. Jack has a question.
Okay.
Thanks.
Okay.
Enjoyed the presentation, Craig. At the one of your earlier signs, you said a healthy soil could reduce phosphorus requirements. Phosphorus is a component of some of our hay meadows, like one in the scene behind me, that we have a hard time increasing those phosphorus levels. So, what can we do in a range or livestock situation, grazing land, to reduce our need for phosphorus?
Yeah, it's a good question, and I looking at this in a. So these are hay meadows. Are they hayed as well as grazed? You were saying, yeah, is that true?
Mm-hmm.
And that's trying to figure out because manure, those are high, how do you cycle back the nutrients into that system, I guess, is the question. I mean, how much is being taken off and how much is being put back? I don't know. I mean, there's not an easy, I guess I don't have an easy answer for you. Trying to understand where it's at, trying to make it more accessible. Because I mean, manure is providing, back into the system. But how much is being taken off with hay, and could you maybe graze when, drop an area off and just graze it versus just hay in it? I don't know. Just trying to think about ways that you could put more more nutrients back into that system without having to provide input.
Okay.
There's getting to be more and more biological amendments that a person can apply, and so, but those, how good they are is always a question. And if I was going to use some of those, I would do small trials on something like that, and because there are phosphorus solubilizing bacteria that potentially could help with that situation, but I would do it on a small scale and just see if they would work on your particular situation.
Thank you.
Any other questions?
If I stop sharing my screen.
You're fine, Craig.
Yeah.
Well, in the absence of no more questions, Craig, thank you very much. This was a fascinating glimpse of soil ecology, and we'll look forward to following this topic in the future. Thanks for joining us today. Thanks all of you.
Yeah, enjoyed it. Thank you. Thanks everybody for showing it.
So long.
Take care, everybody. See you, everybody. Bye.